Let me take this opportunity to introduce Professor Riley, a colleague of mine. So after graduating from Bristol University with degrees in cellular pathology and veterinary science, Professor Riley interned in veterinary pathology at Cornell University in the US and obtained her PhD in immunology and parasitology, subsequently from the University of Liverpool in the UK. She served for 5 years at the Medical Research Council laboratories in the Gambia, and then moved to the University of Edinburgh as a Wellcome Trust Senior Research Fellow. Subsequent to that, she became chair in immunology at the London School of Hygiene and Tropical Medicine. Most recently, Dr. Riley moved back to the University of Edinburgh to take up the role of director of the Roslin Institute in the College of Medicine and Veterinary Medicine. Her research focuses on mechanisms of immunity to malaria, how the immune response can contribute to disease, how immunity affects the distribution and transmission of the parasite, and how malaria infection affects resistance to other infections. In addition, she has a long-standing interest in the biology of natural killer cells and their role in resistance to infection. Dr. Riley is a fellow of the Academy of Medical Sciences and has served on various panels and has distinguished herself with a large number of high caliber international scientific publications. So without any further delay, let me transfer to Dr. Riley, who will give her keynote lecture on malaria, anemia, and invasive bacterial disease. Question is it a neutrophil problem? Professor Riley.
Thanks, Eleanor, and thanks very much for giving me this opportunity to talk. It's a slightly surreal experience sitting in my office at home in the pouring rain in Edinburgh on a cold winter's day and imagining you all around the globe being nice and cozy, many of you. Anyway, some work that we've been doing for about the last 10 years to try to understand a really important clinical problem that occurs in sub-Saharan Africa. And that is that, how do I move these slides? I'll click. Is that what I do? Yeah, okay. So in areas where malaria is endemic, and this is a map of falciparum malaria. So falciparum malaria is the form of malaria that causes the vast majority of the deaths worldwide. It's the most severe. It causes severe anemia. It can also cause cerebral malaria, where children in particular develop comas, go into coma, have fits and go into coma and may die. And it's found throughout the tropical areas of the world, as this map shows. So there's a focus of malaria in the Amazon Delta, in Brazil, in South America. There's falciparum malaria throughout Southeast Asia and the Indian subcontinent, but by far and away the most important continuing focus of malaria transmission, where most malaria deaths happen, is in sub-Saharan Africa. And you can see that the brighter the colours are, the yellows and the reds, that's where we have the biggest problem with malaria currently. Now, what has been noticed for many decades actually is that there's another disease that has a similar distribution in the world, and that's invasive bacterial disease, and particularly invasive bacterial disease caused by Salmonella species. So there are two different groups of Salmonella organisms that we worry about. The first group are the enteric fevers, so typhoid and paratyphoid. And these are particularly common in Southeast Asia and the Indian subcontinent. And they're caused by two particular serovars of Salmonella enterica. And importantly, they only infect humans. So typhoid and paratyphoid are diseases that are transmitted from people to people. My problem is not with those ones in particular. The problem I'm going to talk about are a different group of Salmonella organisms that occur predominantly in sub-Saharan Africa. You can see the distribution is very similar to the distribution of falciparum malaria. And these are the non-typhoidal salmonellas, and I'll abbreviate those to NTS. And they infect a very broad range of vertebrate hosts. Typically, they cause a self-limiting gastroenteritis, which recovers without any particular treatment. But in sub-Saharan Africa in particular, these organisms can invade from the gut into the rest of the body, causing invasive bacterial disease, sometimes called septicemia. And it has a very high mortality, particularly in children and in immunocompromised individuals, for example, those with HIV in sub-Saharan Africa. And in fact, the latest figures that we have suggest that these non-typhoidal salmonellas cause about 30% of all cases of bacteremia in sub-Saharan Africa. That's about 1 million cases a year, and 20% of those cases will be fatal. So that's 200,000 deaths a year due to non-typhoidal salmonella. You might be asking why, as a person who works primarily on malaria, I'm interested in this. And the reason for that is this. So if you look at, this is data collected in the Gambia about 40 years ago, 35 years ago, on the cases of malaria through the year. So this is January, this is December, and you can see in the Gambia, the majority of malaria cases seen in the MRC hospital occurred in the later part of the year, August, September, October. And that coincides with the time of the rainy season in the Gambia, which starts in July, August and goes on until October. And obviously, because mosquitoes transmit malaria and malaria occurs where there are mosquitoes and mosquitoes breed in the wet season, we get this peak of disease. What we also see, for reasons that are completely unexplained, is a similar peak in Salmonella septicemia, Salmonella bacteria, at exactly the same time of year. And this isn't a coincidence, because if we just look here at the proportion of children with sepsis or non-typhoid bacterial sepsis, we see that if they have other kinds of bacterial sepsis, we have a large number of cases there, only a small percent, about 5–6% of those children also have malaria. But we look at non-typhoidal salmonella and we see that actually over 40% of those cases occur in the presence of malaria. And in fact, of all the co-infections we see of malaria and bacteria, 70% of them are due to non-typhoidal salmonella. So this suggests that there is a real problem with these two diseases occurring in the same place. Some recent data from Tanzania shows that as you reduce the numbers of cases of malaria, so this is malaria cases in grey, these grey bars, there have been strenuous efforts in the last 10 years to reduce the number of malaria cases. And in Tanzania, you can see they went from 180 per 100,000 down to less than 20 on this graph. So this is the malaria graph here. So sorry, from 500 cases down to 100 cases in this particular setting. Without doing anything at all to control the incidence of bacteremia, you could see that cases of bacterial sepsis also decreased over the same period, and particularly cases of non-typhoidal salmonella almost disappeared as malaria was controlled. And we've now got data, similar data from several countries in Africa saying as you control malaria, this other disease also disappears, suggesting that they're linked in some way. One of the things that we've noticed is that non-typhoidal salmonella is not just associated with malaria, it's actually associated with anything that causes haemolysis. So human malaria causes haemolysis, that's disruption or rupture of red blood cells, and we see that in human malarial anemia. We also see it in sickle cell disease and in other forms of haemolysis. This particular antibody-mediated one is an autoimmune disease that also causes lysis of red blood cells. And whenever you see this lysis of red blood cells, you also see increased risk of NTS infection, suggesting that there's something to do with hemolysis. Now, one of the things that happens during malaria is the red blood cells lyse when the parasites grow and reach a certain stage. The red blood cells lyse. That releases hemoglobin into the plasma. Hemoglobin is then degraded eventually to produce free heme. Now, free heme is very damaging. It causes oxidative stress and tissue damage. And as a result, it has to be very, very swiftly dealt with by the body. And that happens by binding that free heme to scavenger molecules, which allow it to be taken up into macrophages and degraded. And that happens through the activities of enzymes called heme oxygenases, which break the heme down into three components: biliverdin, which is secreted in the bile, carbon monoxide, which is blown off as you exhale, and iron, which is reused to make more hemoglobin and therefore more red blood cells. In the normal case of just normal homeostasis, day-to-day red blood cells being degraded as they are on a daily basis, there's a constitutively expressed enzyme called heme oxygenase 2. But when there's a hemolytic crisis, such as when you have malaria, then you get an upregulation of an inducible enzyme called heme oxygenase 1 or HO1. And that allows the body to deal with much larger volumes of heme and to convert it to these non-toxic components. So this is a very important way by which the body deals with heme. Just hold on while I just find the right slide. It's not very easy with this system. Hold on. Whoops, all my slides have gone wrong. I'm really sorry. They're all going in the wrong direction. I'm not sure if anybody can help me. Oh dear, dear, dear. Let's see if we can go back. Can we go back? Doesn't seem that you can go back on this system. Can somebody help me to go back on these slides? There's something's happened to the slides.
I don't know. It's Stephanie speaking here. At the bottom of your screen, you should see arrows, and you should be able to navigate the slides by clicking on the arrows.
Oh, okay, right. Okay, let's see. Okay, so you've got me back to where I was. Okay, so let's see if I can go forward. It's not working. Hold on. Okay, that's not working, but the other system is. So let's see if I can do that. Okay. So there's evidence from a couple of experimental models in mice that shows that this enzyme HO1 is really important, and that it degrades free heme, reduces the amount of, so in a model system, free heme would cause disruption, for example, of the blood-brain barrier, allowing inflammation and T cells to invade the brain to cause experimental cerebral malaria. If you not in a normal mouse that doesn't get cerebral malaria, this is the HO1 is produced, and that then prevents the damage caused by the heme. If you knock this enzyme out in this strain of mouse, now you will get experimental cerebral malaria. So we know that HO1 is protective, and if you block HO1, then you get disease. And something's been seen very similarly in another mouse model where the parasite causes liver damage. And again, HO1 is important for preventing the heme from causing liver damage. And if we block HO1, we then get severe liver damage. So there's good evidence that HO1 is protective in malaria, reducing malarial pathology. But there's some other evidence that says that HO1 is very important in bacterial diseases for activating neutrophils to clear the infection. So this is just the hypothesis that we started with, which heme is highly pro-inflammatory, pro-oxidant, and one of the consequences of that is it activates neutrophils and activates the neutrophils to migrate and to undergo an oxidative burst. And this is very important for clearing bacteria. But as I've said, neutrophils and heme are very potential mediators of tissue damage. So this is work that was done by a clinical trainee in my lab, Aubrey Cunnington, who came up with this hypothesis that heme oxygenase degrades heme, suppresses neutrophil function, and therefore reduces pathology during malaria. But the side effect of this, the unanticipated consequence, you might say, of this, is that diminishes resistance to bacterial infection because the neutrophils are no longer properly activated. So Aubrey managed to set up a mouse model to test this hypothesis. And this is the mouse model. So mice were infected with Plasmodium yoelii, which normally causes a transient parasitemia, you can see that here, and clears spontaneously. And these mice become very anemic, so we can see there's haemolysis going on. We can see that there's a loss of erythrocytes during this infection, and we can also see in this infection that plasma heme levels go up. So this is clearly potentially quite toxic for the mouse. We have a couple of controls here. This is phenylhydrazine that causes hemolysis, and that also causes heme to go up. And another control, if we just inject the mice with a synthetic form of heme called hemin, we can also see that heme goes up. So in summary, we have a mouse model where we can raise heme either through the malaria infection or through these controls, and we have an uninfected, untreated mouse here with very low levels of heme. And what we can do is we can then infect these mice with a Salmonella species called Salmonella Typhimurium, which is a natural infection of mice. And we've actually genetically engineered this Salmonella to express green fluorescent protein. And that's what you can see just there. So in this case, we can see that in these mice with malaria, this is different times after infection, the heme has induced HO1 expression. So HO1 mRNA is present in the liver. We can also see heme oxygenase activity in the liver during the malaria infection and after injection of phenylhydrazine. And we can find heme oxygenase protein in the plasma in the malaria-infected mice and in the phenylhydrazine-treated mice. So we can see that we've got induction of HO1. So what we did then was we infected the mice with Salmonella typhimurium. So in a control mice, it is a nasty infection, and the mice will reach the humane endpoint at which we need to euthanize them after about 72 hours. So these are control mice. But the mice that have had malaria or had had heme induced by phenylhydrazine or hemin, these mice all reach the humane endpoint where they have to be killed within 18 hours. So rather than 3 days, we're at less than a day. So this is clearly a much more virulent infection in these mice. When we look in the mice, see the bacterial loads, we can see in the malaria-infected mice, phenylhydrazine in the hemin mice, all of these mice, the bacterial load in the blood, in the spleen, in the liver and the bone marrow is all at least 2 logs higher. So that's 100 or in some cases 1,000-fold higher than it is in the control mice at 18 hours. So clearly these bacteria are growing and proliferating much, much more quickly in the malaria-infected mice than the control mice. And when we look under the microscope at where these bacteria are, so these are neutrophils, and this is a typical healthy neutrophil with multi-lobed nucleus from a control mouse, infected with Salmonella and looked at after 18 hours. And you can't see any bacteria in these cells, and we looked and looked and looked under the microscope and could see no bacteria. These malaria films aren't quite so good, but the phenylhydrazine and the hemin, these are neutrophils, and you can see they are absolutely full of bacteria. These little blue rods are the bacteria, and the neutrophils are absolutely full of them. And in fact, you can see here that not only are the bacteria surviving inside the neutrophils, they're actually dividing, they're actually growing, and you can see them undergoing fission here. You can see these bacteria dividing. So what we think is happening is that the neutrophils are able to phagocytose bacteria very effectively, but are not able to kill them. In the normal mice, the neutrophils very efficiently kill the bacteria and they disappear. Whereas in these infected or the otherwise treated mice, we've got these defective neutrophils in which the bacteria are growing in an uncontrolled manner. So, and here's just an example here. This is looking under the microscope at cells containing the bacteria, and we can do this by counting or by flow cytometry. We can look at the granulocytes, that's the neutrophils, and also the monocytes, the macrophages. And we can see whether the mice are infected with malaria or not infected. The proportion of cells that contain phagocytosed green bacteria, you can see an example there, is not different. So after 45 minutes, the cells have phagocytosed the same number of bacteria. But if we look at viable bacteria, that's done by colony counting, we can see after 45 minutes, there's no difference, the same number of viable bacteria. But after 2 hours, the uninfected mice are starting to kill. They've reduced the number of viable bacteria by more than half, probably by about 80%. This is a log scale. But the malaria infected mice have not killed any bacteria at all, as far as we can tell. So we think the bacterial killing is defective. And we can look at that again by this assay looking at the respiratory burst, the oxidative burst. We take whole blood from the mice or bone marrow from the mice. We stimulate it with a mitogen, a calcium ionophore PMA, and that induces the oxidative burst, and that converts a non-fluorescent substrate, dihydrorhodamine, to fluorescent rhodamine. And we can see this by flow cytometry. So GR1 is a marker for neutrophils. So this gate here in the flow cytometry, those are our neutrophils. And this is the rhodamine marker, the fluorescence. And if we don't stimulate the cells, we see really no cells that are fluorescent. But if we stimulate the cells with PMA, we get this population here that are rhodamine fluorescent granulocytes. And these are the granulocytes in which the oxidative burst has been induced. So we can look at the number or the extent of fluorescence here to see if the oxidative burst is normal or not. So here's an example. This is our control mice, and this is the oxidative burst set to a nominal value of 100, and then we're comparing with that. So this is normalized data. And you can see in a malaria-infected mouse at day 7, day 14, or day 21, the oxidative burst is at best half as good as it is in a control mouse, and at worst it's suppressed by about 75%. And we can see the same thing in the bone marrow. It's not quite as obvious in the bone marrow, but it's certainly there, with a reduction by about half of the oxidative burst in granulocytes or neutrophils coming from the bone marrow. And is this due to HO1? Well, the easiest way to test that is to block HO1, and we can do that with a competitive inhibitor of HO1 called SNPP. SNPP binds to the active site of HO1 and blocks its activity. So here's a normal mouse, and here's the bacterial load after Salmonella infection. And here's a mouse just treated with SNPP. So the SNPP itself doesn't do anything to the bacteria. Here's a malaria-infected mouse with a very high bacterial load because its neutrophils are defective. If we block the effect of HO1 with the inhibitor, we can significantly reduce the bacterial load. And in fact, the bacterial load here is not significantly different from in the control animals. The treatment, the SNPP, has no effect on the parasite growth, so it's not affecting the malaria infection. And we can actually see that it reduces the number of granulocytes that have got bacteria in them. And we can also see here that it reduces, these are our granulocytes, and this is GFP. So these granulocytes have live bacteria inside them, the GFP Salmonella. And when treated with SNPP, these mice don't have that population. So we're pretty confident now that this whole pathway of impaired neutrophil function and inability to control the Salmonella infection is due to HO1. Now, those of you that are interested in global health will be saying, that's all very well, that's humans. Do we have any data? That's mice. Do we have any data at all that we see something similar in humans? So if I can just move the slides on a bit. So this is data from the Gambia in West Africa, comparing children with uncomplicated malaria. So children who turned up at outpatients and were treated and sent home. So you can see their total plasma heme on the day they were first seen was quite variable. In some of them, plasma heme was quite low. In some of them, it was very high. When we saw them a month later, plasma heme had returned to low levels in most of the children. So you can see that these children in particular have very high levels of plasma heme. If we look at the plasma HO1, each of these dots is one child, you can see that these are healthy controls. So normally we don't find any HO1 in the plasma. But in these malaria infected children, we see quite high levels of HO1 in all of the children. And when we look at their neutrophils by flow cytometry to see if they have the correct oxidative burst, we see something very interesting. So let's look at this one first. This is the healthy controls. These are healthy children, and this is flow cytometry. So the further they are over here, the stronger the oxidative burst. And we have this population of neutrophils here, which represents 90% of the cells that have what we call a normal oxidative burst. And we have a small population down here that are maybe not very good. And they're probably immature cells that have just come out of the bone marrow. If we look at the children with acute malaria, we see that about 30% of their neutrophils look normal, but 70% of their neutrophils are defective. And their oxidative burst is about only 10% of the value of this one. When the children have been treated, so a week later, there's actually not much difference. Still 30% normal, 70% abnormal. A month later, still 30% abnormal, 70% abnormal. 2 months later, this is 8 weeks later, we're just about back to the healthy controls, and this one looks quite normal. That's just data from one child. If we look at data from all of the children, we see this. So this is the rhodamine expression. The higher it is, the better. These are our controls. At day 0, day 7, day 10 to 8, we have this group of children here with really defective neutrophil function. And it persists for a long time, at least a month, and in some children 2 months after their malaria infection has been treated, their neutrophils are still defective. And I think you can see that if their neutrophils are defective for weeks or months after their malaria has gone away, those are the children that we think are at risk of getting invasive bacterial disease. These children with defective neutrophils, I predict, are at very high risk of getting bacterial infections. So this slide here just summarizes everything I've said. Malaria causes the breakdown of red blood cells, release of heme. Heme is detoxified by this enzyme HO1, and in some way that we don't understand, this causes the granulocytes to be defective. And that, we suspect, is what causes the increased risk of bacterial disease. So how can we use this? It's possible that we could use these HO1 inhibitors to reverse the effect. But to be honest, I think in a clinical setting that's unlikely. This inhibitor has been trialed. It is safe. It might be used. My guess is that rather than that, we may be able to use this very simple flow cytometric assay of neutrophil oxidative burst to identify children who are at risk of bacterial infection after they've had malaria or anemia, for example. And if we see this defective oxidative burst, we may decide that it's a good idea to put these children on broad spectrum antibiotics for a period of weeks until their neutrophils recover. another piece of work, which I don't think I'm going to have time to talk about because I can see my time is up, is that there's a lot of persistent subclinical malaria in sub-Saharan Africa. Children who are clinically quite well, who are going to school, playing as normal, but are actually carrying malaria infections. And what we've seen in some pilot data, and you'll be able to see these slides later for yourselves, is that actually these children with these chronic malaria infections do have raised HO1, do have high levels of heme, and we think that this may also be a risk factor for bacterial infections. And that's a piece of work that's ongoing. We're quickly just doing some work in Burkina Faso to look at chronic malaria infections, and we can see that these children, let me just see if I can very quickly whiz through this, that these children have high levels of these are controls, high levels of HO1, and I'll skip through that. Early analysis of this data says that asymptomatic malaria infection leads to raised heme. This leads to raised HO1. They also have high levels of IL-10, and that in itself can drive HO1. So we're now carrying out a study to look at the consequences of this in children with subclinical malaria infections that are not usually treated. And I'll stop there. I think I'm more or less on time. I want to thank all the people who've helped in this work. Aubrey, whose work it really is, who then he obtained his PhD and is now a member of staff at Imperial College. Collaborators in Nijmegen on the subclinical malaria. We've got a clinical trial going on in the Gambia at the moment with the director there, Umberto D'Alessandro. And I have a small group of people in Edinburgh, Jason in particular, who's the postdoc, who's leading that work in the Gambia at the moment with Umberto. And I'm going to stop there and see what happens next. I hope that made sense, and I hope you could see, hear and see everything you needed to.
Thank you very much, Professor Riley. There are a couple of questions perhaps to respond to. Is that okay?
Yes, absolutely.
Okay, so the questions that I've received so far in our chat line, first one is this neutrophil defect reversible or is it permanent? Or how long lasting?
Okay, so we see that it can be quite long lasting. We think it is reversible with time, but it takes a bit longer than you might expect. Anybody who's listening who knows about neutrophils, the story about neutrophils is that they're supposed to be very short-lived and that they sort of turn over in the blood every 24 to 48 hours. Our data suggests two things. Either they're longer lived than we think, and actually there are some data now from mice suggesting that unactivated resting neutrophils do indeed have a much longer lifespan than we think. The other possibility, which I think is a strong possibility, is that actually the effect that we're seeing is not on the circulating mature neutrophils, but is an effect on developing neutrophils in the bone marrow. So that we think there's something gone wrong with the neutrophil precursors in the bone marrow, and that those precursors keep turning out defective neutrophils for a period of weeks until in some way either the precursors recover or whatever it is that's affecting the precursors disappears. And one possibility that we know, when red cells break down in a malaria infection, the heme gets incorporated into a very, very long-lasting pigment called hemozoin. Hemozoin sits in macrophages, and it sits in macrophages in the bone marrow and is degraded very, very slowly over a period of weeks. And one possibility is it's this long, protracted degradation of hemozoin in the bone marrow that is continuing to induce HO1 and continuing to affect the neutrophils, so that they come out of the bone marrow as a defective cell for many weeks until finally all of that hemozoin is degraded. That at the moment is just a hypothesis, but it's something that we're looking at.
So the other question that I got here before me, could it be that this malaria-induced arginine deficiency is involved in this mechanism of...
So we haven't looked at arginine in particular, and I don't know the answer to that question. The other thing that people do sometimes ask is whether, because we're releasing a lot of free iron, we know iron enhances bacterial growth, And I think that's also a possibility, that one of the things that's happening is that the heme oxygenase is producing a lot of iron, free iron, and it's that iron that is helping the bacteria to survive. We've got some experiments going on to look at that as well at the moment.
Another quick question. You mentioned Salmonella. Are there any other bacterial infections where this plays a significant role?
Yeah, so we're pretty sure. So we see other gram-negatives that have a similar effect. I don't think it's specific to Salmonella. I think we particularly see Salmonella probably either because they're already present in the gut and therefore are in a position to invade when the opportunity arises. Or maybe because there's something else going in the gut that is particularly favouring invasion of Salmonella species. And we know that there is a little bit of gastrointestinal inflammation and dysbiosis in the gut during malaria infections. Whether that is in some way favouring Salmonella to grow, we don't know, but we've got a little bit of data on that. What we don't think is happening is we don't think it's just that they're catching Salmonella in their environment. There have been some studies to look at whether children who get invasive Salmonella disease are catching it from their local environment, and there's no evidence that that's happening. So we're not quite sure why it's Salmonella, but I think it's also the case with other bacteria.
All right, so I think in the interest of time, I'll have to wrap up here. Thank you once again for a great talk. If you do have time on the chat line, you'll see there are a number of questions, and if you can respond to them, that would be awesome.
I'll see if I can. I'll see if I can master the technology to do that.
All right. Thank you once again.
Okay. Thanks a lot.